Hi, everyone. Thanks so much for coming today. We're really pleased to have this seminar really designed as to how we can promote better collaboration and interaction um, with our industry colleagues through a really interesting pathway that we like to call the investigator-initiated studies pathway. It's typically a robust program within the biotech industry, making available their products or devices or diagnostics for additional research use, led by some of our faculty. Just for background, I'm Grant Wells, Director of Innovation and Development for the Maternal and Child Health Research Institute here. And I'm pleased today to be joined by Professor Greg Enns of Medical Genetics here in our Department of Pediatrics. Greg has just a wealth of experience working with industry, participating in data safety monitoring boards, leading registry studies, and collaborating and discovering new treatments. He's also an entrepreneur himself, which he'll probably tell you more about. And then also a wonderful colleague, Tran, who is Executive Medical Director at Astellas, leading a lot of their clinical development initiatives. She keeps adding more and more to her plate, and her role changes by the day. And it's just wonderful to see all the really interesting things that she supports at Astellas, but also is a great partner to us in a adjunct clinical capacity where she's coming alongside our faculty as well as Jessica and I to kind of help support more of our translational and clinical trials interests. So like to pitch this over to Greg in the interest of time, and Greg, take it away.
All right, thanks so much. So I greatly appreciate the opportunity to talk, and thank you, Grant and Jessica, for inviting me. Originally, I wasn't sure what I was going to say, but as I thought about it, I think I have done quite a bit with industry, and I'm happy to discuss things with the topic really focused on investigator-initiated studies. Dr. Tran is going to be the person who will really provide further details on how this works through the industry side. But I thought I would give a little bit of background from an investigator side. So to start, here's my conflict statement. You should know I am a co-founder along with other Stanford faculty of a new enterprise called Evia Therapeutics. And I've consulted with a number of companies who are working on developing therapeutics, especially for rare genetic disorders. And these are companies working in the fields of either small molecules or even messenger RNA and gene therapy. But a lot of things are being developed. As mentioned, I also serve on data monitoring committees as an important process of the interacting with industry as they're bringing therapeutic products to the bedside. And as a co-PI on a variety of different clinical trials in the field as well. And as Grant had mentioned, patient disease registries are also part of what we do here. And it's important to understand the natural history of these conditions so we can design better therapeutics and understand what their effects are on potential outcome measures. So a bit of background for me, I tell people I'm a biochemical geneticist, and most people go, okay, well, fine, whatever that is. So what is it that I do? So I did train as a pediatrician at Children's Los Angeles, and then I did further training in medical genetics, also called clinical genetics at UCSF. Following my genetics training, I also spent a further year focusing specifically on biochemistry and biochemical genetics. So like most geneticists, detective work plays a role, but here detective work typically is in a laboratory with a machine like a tandem mass spectrometer here at the bottom, which happens to be named Sherlock. This was our first tandem mass spectrometer. This is in Tina Cowan's laboratory and has served us well throughout the years, but it's using a machine technology in order to diagnose patients with these rare inborn errors. I show a picture of Bob Guthrie here, who pioneered newborn screening, and a lot of what we do is newborn screening based because we want to identify children before they exhibit symptoms so we can treat them pre-symptomatically. Of course, showing my like of Star Trek, designing better therapies, looking towards the future, and trying to figure out how to take care of rare disease patients is also what I do. So to start, clinical research in opposition to what's basic research is listed here. And basic research, of course, is something that's performed not necessarily with a practical end involved, but it's something that's done to understand nature and its laws. But in 1997, the NIH Directors Panel on Clinical Research issued this three-part definition where clinical research consists of either patient-oriented research, epidemiologic and behavioral studies, or outcomes research and health services research. What we're really focusing on today, of course, is patient-oriented research, because this is involving human subjects and it's involving our patients, and it might have direct influence as far as a specific treatment or therapy, or it might be just material that we're using to develop things like biomarkers or other types of diagnosing individuals who have a variety of different conditions. So these are people who an investigator or colleague directly interacts with. And the patient-oriented research, as we all know, focuses on either mechanisms of human disease, therapeutic interventions, including clinical trials, and as well, importantly, the development of new technologies. This is sort of morphed into what's called translational research. And I was interested to find that in the 1990s, translational research was first really mentioned in the medical literature, and it really surrounded the cancer field. And it referred to work spanning different types of research, for example, immunology studies spanning basic and clinical research, or work spanning disciplines within a particular type of research, like bench research involving molecular genetics and immunology. So it's sort of morphed into what we currently consider to be translation. And this was defined by a recent report published by the Evaluation Committee of the Association for Clinical Research Training, stating that translational research fosters the multi-directional integration of basic research, patient-oriented research, and population-based research with a long-term aim of improving the health of the public. And importantly, translational research moves in a bidirectional manner from one type of research to another. For example, from basic research to patient-oriented research, or population-based research, and back and forth. It involves, importantly, again, collaboration among scientists from multiple disciplines, shown in the cartoon here. And really, a basic summary of this is from bench to bedside. And I might add, and back again. So this is a process that goes back and forth from designing experiments and seeing how these experiments come to play, or clinical trials come to play in our research, and then have that research inform further studies that can advance the field. When speaking of investigator-initiated studies, one form, of course, is the clinical trial. But unlike the sponsored research or research that is performed in the auspices of a industry partner providing a clinical protocol and the site PI agreeing to that protocol and participating as part of an investigator in a typical multi-system or multi-center trial, an investigator-initiated study is a study that is conceived by the investigator themselves, and the research idea, the study protocol, and ultimately even being the sponsor investigator sits on the shoulders of that individual, which I really truly found daunting when I first was considering doing such investigations. And I could not do it without MCHRI support, I can tell you from the start. If the investigator typically would select a qualified team, would be responsible for overseeing the IRB protocol submission. And once this has been created, of course, coordinating and conducting the study would be performed under the investigator's view. So continuing to have important emphasis on protecting the rights and safety of participants, performing the informed consent process, often with the help of the study coordinator, and in my experience, often supplied by MCHRI. Monitoring the investigation, of course, dispensing the drug or the investigational device, And also importantly, interacting with the FDA. As an investigator, I have to play the role of the sponsor, and I need to formally request an IND for a new product from the FDA related to the biologic drug or the device that I'm interested in. And I also have to write periodic reports in order to document the use of the investigational product. In addition to clinical trials, there are other translational studies that I certainly have participated in and do not necessarily have to do with the interaction of a company or a sponsor providing a drug or a therapeutic. And similarly, in these situations, the investigator would conceive a research idea and develop a study protocol or methods, typically also needing IRB submission because it's dealing with human subjects and generalizable knowledge. And the investigator would have a role in creating, coordinating, and conducting the study, just like anything. Similarly to the clinical trials, informed consent and protection of the rights and safety of participants are paramount. The investigator, of course, would monitor the investigation, analyze data, and report it ultimately. And this is importantly very much a team approach typically. When thinking about funding, I'm not really going to spend any time on NIH. That's for probably another day, but just to be complete, the NIH, of course, has a variety of different programs that are focused on translational research, including the National Center for Advancing Translational Sciences, or NCATS. The Clinical and Translational Science Awards Program is something that has been done under the NCATS umbrella, and a variety of different grants, such as small business grants that can be used to receive funding for translational research. But talking today, we're really going to focus on industry. And I'm also going to mention, just because of my own experience, some foundations and internal funding, of course, are other ways to get support for an idea. And in my practice, it's been a patchwork quilt, a little bit of everything. And some of the studies that I'll talk about as examples, you'll see that I had some funding from a foundation, also some from internally, like MCHRI funding or Spark funding, and sometimes no funding at all, but with goodwill and a fellow and some clinical research, a little bit of elbow grease gets the job done as well. So common themes that you should probably be expected to find when applying for an investigator-initiated study are listed below. I'll mention them briefly. A lot of these studies, you can find an online application simply by Googling investigator-initiated study plus the company of interest. Oftentimes the company or industry partner will have an internal review committee, and they're really variable review times. One example from a company that I've worked with has monthly review committee meetings, but investigators or interested applicants can put in an online application at any time. The applications vary in their rigor, I would say, as far as the amount of detail that needs to be provided. Sometimes they're really quite brief, other times quite detailed, like an NIH application. But research background and design are typically part of these applications. It's important to note that it would be helpful, I think, if you look at the website, because oftentimes the sponsor website will have key areas of interest listed as far as what they're encouraging further studies in. But you also might have an idea that has the potential interest to the sponsor who's not necessarily using a medication in a certain disease area. Research background can also include disease registries for natural history, because as I said, natural history is so critical to understand these disorders in order to understand how therapies work and whether we're actually helping with such treatments. Typically, the studies do not support animal model work, but that's, I'm sure, going to be variable depending upon the sponsor. A statistical analysis plan is often required and would be expected, so you could justify your sample size and what you're going to do. The overall research plan, budget, conflicts of interest statements, etc., are all common. I'm also including just a brief note on foundation as an example. This is a foundation I've worked with before, and just to give you an idea that sometimes they want things to be really quite concise. And I thought this was a nice example of that, where on the website says grant proposals should demonstrate the impacts and value to the people served. Is this really meeting the goals of the foundation? And clarity in writing and short, concise proposals are more effective. Brevity is an asset. And that's something that I've certainly have found. I tend to be verbose, thinking about NIH applications. I might add things quite a bit as far as experimental design and things like that. But oftentimes, less is more when applying to a foundation. So in just a few minutes, I'm not going to take too long, just so we can have Dr. Tran also give her a talk. I'm going to go over some examples of clinical trials and other studies that I have done over the years involving both medications to study subjects, and also the development of a variety of different translational products, so to speak. So biomarkers, especially, and exploring novel therapeutics, especially AMP-K agonists or AMPK agonists, which we've been using to treat mitochondrial disease. And finally, a little bit on solid organ transplantation as a therapy for our inborn error patients. So triheptanone is an example. This is a medication that is used now and approved to treat long-chain fatty acid oxidation disorders. And these patients typically present with a Rye syndrome-like illness, hypoglycemia. They can get into real trouble with cardiomyopathy. And the manufacturer of this medication did not have in their clinical trial a type of patient called translocase deficiency, which is another fatty acid oxidation disorder that this medication should have an effect for. So I actually had to request it directly from the sponsor to give this medication to me. This was a little bit unusual because this was an emergency type situation where I requested this urgently and emergently, contacted the FDA, got an urgent IRB approval, and within 48 hours, I think, between the conception that we might be able to need or use this medication and get it for our patient, we were able to initiate the study with IRB and FDA approval. So these things can go together pretty quickly. And this was a medication used in a patient in the PICU who had fatal cardiomyopathy developing and had an ejection fraction below or near 25% at the time of starting the therapy. And within 3 days, the ejection fraction had improved to 75%. So it's a solid, nice outcome. And this was used again in other patients who had this rare translocase deficiency. And we were able to report our results. So that's one, maybe an emergency example. This is not so much an emergency, just the opposite, as a matter of fact, where this is a medication, a quinone, that we've been using to treat mitochondrial disease patients. And this medication called EPI 743 was initially approved only for patients who had mitochondrial disease, who are considered to be within 90 days of end-of-life care. So very severely affected patients who are on the decline. This was an investigational, it was a new drug, and the company was allowed to supply it to only patients who are most severely affected. I requested and kindly was given this medication to also use it in patients who have more mild conditions or even secondary mitochondrial dysfunction. But that was not so much an urgent care, but this was gratefully received. And something else, just as an example of where you can expand into with a company. Also, my colleague, Francis Blankenberg, worked on, as an aside, another part of this story relates to biomarkers. And the company was interested in treating mitochondrial disease, and we were having patients coming into our center for mitochondrial disease therapy. and Dr. Blankenberg had an idea for how to monitor these patients. And this was not part of the clinical trial protocol, but this was an investigator-initiated study where we were seeing patients who were being treated with a novel compound, and we wanted to be able to measure this better. And Francis was able to develop a protocol and a technique using a technique called HMPAO spectroscopy, or SPECT, that was able to measure the mitochondrial function or the redox state of tissue directly. And these are sort of before and after shots of different parts of the brain using Dr. Blankenberg's technique that showed that after treating with this medication, the redox potential, the glutathione levels actually improved in these patients. So this was a way to measure effects of the drug that was not really part of the initial study at all. We also have an interest just in our group in measuring blood for biomarkers in our mitochondrial patients. And typically, these are not necessarily biomarkers that are measured for safety or not typically used in common, because these are novel biomarkers that are not necessarily commonly used in the clinic today. But because of support given from foundations and MCHRI funds, we're able to discover that the glutathione is actually a very good biomarker for our mitochondrial disease patients. This is just showing a shot of the difference between the glutathione redox potential in normal subjects versus controls, showing that there is a real significant shift in redox potential, meaning basically our mitochondrial battery is going down when we're sick if you have mitochondrial disease, and the sicker you are, the lower the battery gets, and we can measure that by measuring glutathione. But this work actually started, and it was my first grant I ever received as a academic physician, and that came from the United Mitochondrial Disease Foundation. So it was a foundation. We got further funding from MCHRI and continued our work. And finally, just in brief, I wanted to mention AMPK, because this again sort of is a study that arose from our work on biomarkers and measuring mitochondrial function in patient cell lines. And through MCHRI, I was able to get funding to support a fellow and a research scientist to work on these cell lines. And one research scientist in particular, Teresa Moore, came up with an idea based upon some of this work that an AMPK agonist could potentially really have an effect for mitochondrial disease patients. She was working with a known drug called PT1, which is being used in clinical trials for diabetes, but also is noted to have specific function as a AMP kinase activator, and nicely showed in cell lines and then animal models that this compound and others like it improved mitochondrial function and improved outcomes as far as eye disease that are related to mitochondrial dysfunction, and potentially even brain disease, because following this initial work, which was also supported by foundation and other grants such as the Tauber Foundation, we're able to then develop our own products and develop AMP kinase activators that specifically cross the blood-brain barrier and stimulate AMP kinase as a unique first-in-class medication. And that led to recently, over the last couple of years, the founding of Evia Therapeutics, which is a spin-off company based upon this work that is really focusing on activating AMP kinase to treat mitochondrial dysfunction. And this can range from anything from pediatric mitochondrial disease, the patients that I see all the time, to the eye disorders and disorders such as Parkinson's disease. So that's been exciting for us. And I don't want to forget elbow grease. And this is work that Anna-Kaisa Niemi did, working with Carlos Esquivel and his team in liver transplantation, really pioneering the transplantation for inborn errors of metabolism, especially organic acidemias and urea cycle defects, getting the bandwidth. And during their training, we've had various fellows who have worked very hard to get things published that were basically elbow grease, and going through the IRB process, going through discussing things and consenting patients and getting things done and looking at old data and data on the shelf, so to speak. So there are many ways to go forward. With that, I'm going to end. These are resources that I found useful. I think this is going to be a lecture that's going to be available for future viewing, so I won't keep that up long. And of course, as emphasized, this is a team sport, and everybody needs to collaborate. And I certainly have many people to thank who have been our collaborators throughout the years. So with that, and not wanting to take too much more time from Dr. Tran, I think I'll end and we can move on. Thank you so much.
Thanks so much, Greg. Really appreciate your time. And if anyone has any questions, we can put those in the chat. Maybe take a couple minutes here while we wait for Ha to kind of queue up. Or if not, we can have Q&A all the way at the end. Excellent. Okay, I don't see anything in the chat. Ha, take it away. Thanks so much for being here.
Oh, thank you for having me. Yeah, special thanks to Grant and Jessica for inviting me. It's funny, I was reflecting on my career, and 10 years ago, I was applying for my first MCHRI grant, and I was a second-year fellow at the time. And now 10 years later, a lot has changed. And Dr. Enns, thank you for your talk. I think this is the importance of having like in-person connections, because how we came up with this idea was just everybody, Grant, Jessica, myself, and Dr. Enns running into each other in the courtyard. I'm like, hey, let's do a talk. With further ado, my name is Tran, and I have been with Stanford for the past 11 years, as initially as a pediatric nephrology fellow, and then I stayed on initially as a clinical instructor for a few years until I was more immersed into pharma, and then I became an Adjunct Clinical Assistant Professor, in which I volunteer my time. So currently my day job, I am Executive Medical Director at Astellas Pharma, which is a mid-size and moving to large-size pharma company. It's a global company. We actually have a headquarters here in San Francisco with the acquisition of Audenti's gene therapy, which is now Astellas Gene Therapy. So I oversee rare disease and gene therapy programs. I started my career in industry almost simultaneously, I think, as during my fellowship. So during my fellowship, I had the opportunity to pursue the master's in epidemiology and clinical research, which I did here at Stanford, and then realized I really wanted to work in clinical trials. I then proceeded to do a one-year fellowship at Genentech Roche, and then stayed on at Genentech Roche for an additional 4 years, working across both pharmacovigilance and clinical development. Afterwards, I spent 3 years at Baxter International, which is mainly a medical device company, but we did drug device combos, for which I led the clinical development for the renal care franchise. We did, during my tenure, did achieve a first-in-class FDA approval for a novel dialyzer, and additionally, emergency use authorization EUA for a sepsis filter. About a year ago, I returned to pharma and joined mostly I'm very interested in the pediatric development that was ongoing there. So for disclosure, I am employed by Astellas, and the views that I share today are not specifically of Astellas or Stanford University, but they do represent industry standards. So again, wanting to talk about the importance of pediatric research. So without pediatric development, there is no adult product, and this is investigational medicinal products. So when you have a marketing authorization application, even if the indication is, something like prostate cancer, where it's like purely seen in adults, you still need to have a pediatric investigational plan to study the pediatric patients, a waiver or deferral to be in place. And some decisions that are made early in development can have profound impacts on future requirements for pediatric development. And examples of this is, when we're planning for, the life cycle of the molecule and our clinical development plan and our evidence generation plan from the industry side of things, you know, if you decide early on without doing your research that you're not going to pursue pediatrics, that is can have a major impact, especially if you go back to the regulators later and they ask you to study the pediatric population. And what I'm speaking about here is in terms of, you know, needing to do special formulations for pediatrics. Like oftentimes the peds population may be unable to take pills, right? So you may need a liquid formulation, you may need to have a G-tube formulation, and then you would need to test those out in terms of G-tube compatibility. Will it stick? Will it clog the G-tube, etc. So the paradigm of drug development has changed. So previously, you know, everyone thought of adult development first. Let's get through the adult program and then we'll think of pediatrics. However, that has changed significantly over the past decades. And current status is really thinking about the adult and pediatric development at the same time at the point of what we call candidate selection. So typically as a drug developer, you know, we have things coming down the pipeline, often like an idea, an in vivo testing, and then testing in animal studies. And when you get to a point where you've achieved your you know, mechanism of action in animals, and you think through, you know, which indications you want to go in, really this should start in terms of, you know, adult and pediatrics, adult only, etc. So just a brief overview in terms of the pediatric regulatory requirements. So in the EU, there is no exemption for orphan drug designation. And I talk about orphan drugs because a lot of what rare diseases do affect the pediatric population as well as the adult population. And then in the US, there is no specific pediatric requirement for orphan drugs. So just to give an overview of what this means is that, you know, the FDA really wants to prioritize development and evaluation of new treatments for rare diseases. And so they have the authority to grant orphan drug designation to a drug or biological product, and even devices to prevent, diagnose, or treat rare disease. And how they define it is overall like an estimated prevalence of less than 200,000 patients. And then for orphan drug in the US. And then the reasons why oftentimes companies will look for orphan drug designations is that they can qualify for certain incentives, such as tax credits, exemptions for certain fees, and then potential 7-year market exclusivity after approval, which is really a big point here. So basically market exclusivity means that even though there may be other similar products coming down the pipeline from competitors, you know, they wouldn't be able to market that for 7 years until the first to market gets there. Okay, and then I won't go over too much in terms of Europe since we are here in the United States, but just kind of an overall to understand that in Europe, they do give a 6-month extension for protection. They have 2 years of exclusivity for orphan products, and then also get this pediatric use marketing authorization. And in Europe, their fines are in the US, there really aren't too many fines. They can have a refusal to file, but Europe is actually quite more heavy-handed when it comes to penalties, and that includes financial penalties. And they also have like a public name and shame list where they do list the names of companies that were not compliant with pediatric regulations. And this shows the evolving landscape in the US as well in terms of the prioritization of pediatric research. So in 2002, the BPCA, or Best Pharmaceuticals for Children's Act, they made it voluntary for pediatric drug studies, and they reauthorized this pediatric exclusivity provision. This changed a year later with the Pediatric Research Equity Act, where it allows the FDA to require pediatric studies in drugs and biologics for certain applications. And then most recently, there were some changes, and I think most prominent here is that they have eliminated the orphan exemption from pediatric studies for cancer drugs related to relevant molecular targets. So the FDA has a list of molecular targets that either are not relevant to pediatric research as deemed by the FDA or relevant. So what they don't want is they don't want companies to apply for exemptions based on molecular targets, not based on the indication alone. So in terms of like timing of pediatric programs with the FDA and EMA, typically, the PIP is the Pediatric Investigational Plan, which is required by the EU. So the companies will typically think about their strategy between phase 1 and phase 2 study. And per regulation, by your end of phase 2 meeting, before you go to phase 3, you really need to have your pediatric investigational plan submitted. So typically in a pediatric investigational plan, you have, whether your request for either deferral of studying pediatric patients until you've established safety and efficacy in the adult population. I think this is most often the most common ways that companies do look at pediatric research, and then they'll start their pediatric studies after phase 3. There's also a waiver, which is the time to ask for one. Let's say, for example, there are certain diseases that you don't really see, and then usually the age groups are the neonates, so less than 28 days. You can ask for a waiver there, or you could less than age 2, less than age 6, or if you want to only study adolescents up. And in terms of how you receive your waiver, typically, most often you have to demonstrate through like epidemiology studies or whatnot that either the disease doesn't exist in that pediatric age group, or that is excessively rare and that it would be not feasible to conduct this study, or if there's any safety reasons based on your animal or juvenile tox studies. I think oftentimes the regulatory authorities don't like it too much if you talk about like feasibility, right? But in terms of safety, that's one component that is acceptable. And also another component is if it really does not exist. Typically, most companies will have your PIP, this is the EU, and the FDA has something called a pediatric study plan, and you try to go align and go in at the same time. That's the most efficient way to go about it. Sometimes things will split off depending on if it was a US only study or whether it's a global study. I know Dr. Enns already reviewed the definition of an investigator-initiated study, but just to repeat, from my end, I really wanted to say that, this is a study that was conceived by the investigator, and it's not related to any, it's not without any influence, I would say, from the industry sponsor. The FDA also has a definition as well, is it's the individual who initiates and conducts the investigation, and whose immediate direction of the investigational medicinal product is administered or dispensed. And then in how it works is that typically this independent investigator will make a request to the company to obtain the investigational medicinal product, or maybe they want the drug only, or do they want product plus extra funding or extra support, and then the investigator would be the one to conduct the clinical study. So what's needed at a minimum, right, in order to start this process is the investigator name and institution, study properties, including objectives, target population, or animal models or design. And Dr. Enns, we do, I think it does depend on the company, but my current company does accept applications for animal studies as well, and also the nature and quantity of the involved company product. I think what's interesting here is that there are certain universities that are very, very good at requesting product from companies or requesting funding to carry out certain studies, including animal studies or pathology studies, or even radiology studies. So here's an example, like introductory note that oftentimes I would receive or other members of my team would receive. So you would have like, dear pharma company, my name is Dr. X. I'm a pediatric specialist at Stanford University. I noticed in your pipeline, your article, your presentation at a certain Congress that you're currently studying the use of, whatever study drug for unicorn horns in patients ages 18 and up. Over the past year, I've noticed an increase in growth of unicorn horns in our practice. And you can state, I think at Stanford, typically you guys do see, we do see patients up to age 26, since there are no approved, limited treatment options to treat unicorn horns in the pediatric population. I hope that we can schedule an initial conversation to potentially collaborate in generating useful data for the patient and research community. So this is something that I would typically get from a university member. And then, what would be submitted, and this is from the company side, is like what we look for. So usually a cover letter. After having that initial conversation, we talk through like what's necessary. So a cover letter with your scientific question and hypotheses, confidentiality agreement. So this can be initiated by either the company or by the institution, such as the university. And then the study proposals, again, I agree with Dr. Enns, it does depend on the company. So the previous company I worked for, I was fortunate to be able to collaborate with Stanford and provide them with a $100,000 grant for health equity research. And it was literally a page and a half of what they needed to do for their synopsis. So this is my current company where they asked for like 10 to 15 pages, but draft protocol or protocol synopsis, the title, the hypothesis, objectives of your study, background and significance, the design, the phase of development. Is it a phase 1? Are you planning to do something larger? Is it single center? Is it multi-center? Description at the IMP, including dose strength and formulation. That's something you probably would collaborate with the company on. Your duration of the study and your statistical analysis plan, including sample size justification, power analyses. Additionally, need to demonstrate the expertise of the study team, references, and love references. Like if you have something that previously shows your research or abstracts, that would be great. Budget. I think this is something MCHRI can help with. Estimated amount of IMP to be supplied by the company. So again, how often will you be dosing the patients and for how long? The CV of the investigator and the sub-investigators, conflicts of interest. Again, is this a single center US only study, or are you planning to collaborate with globally? And then again, you need to document your IRB approval here. And then in certain cases, like Dr. Enns mentioned, if you're submitting an IND to the FDA, definitely need to document that. From this company side, what we take into consideration for funding, again, number one, this needs to be an idea that was initiated by the investigator and not solicited by the company. So we don't go out and solicitate for people to apply. And then, we do look at the overall strategy for the product. And then any consequences that could be going on in terms of like safety signals, scientific merit and rigor of the planned study. Is it appropriate for the clinical development plan? And I think this is something that's important. So for an example, at Astellas, one of our largest projects, what most of you are probably aware of, is Prograph or tacrolimus, but it's been on the market for, I don't know, at least 20, 25 years or so, like very long time. And so, would it make sense to start a study now for Prograph? Likely not, because it's at the end of its life cycle. Again, it's still a very important product. It definitely differentiates in terms of the brand versus generics. We don't have much generic erosion, but at the same time, a product that is at the end of its life cycle may not have as much investment as something that is in phase 1 or phase 2. Again, safety monitoring and reporting of toxicities, these things all get put into play, and then appropriateness and availability of the budget. I would have to say that Stanford has the highest budget, I think, in the probably in all of the US, except for maybe CHOP. So Stanford is pretty high up there. So that's also something we do take into consideration. And then the overall budget and resources that it would take from the company in terms of, manufacturing and labeling, supply, et cetera. And then we also think about the availability of the investigational medicinal product. So again, like if you see something that the company has in their pipeline, let's say there's let's just pretend like there's a new viral illness out there, and there's a vaccine that you want to test in a pediatric program, but do they have a pediatric dose, or do they have the formulation available? So this is also something the company needs to think of. Is like, yes, we would really love to study this in the pediatric population, but maybe it takes 1.5 years. That's typically the turnaround time to get it to that formulation, to test it, to make sure that it's safe and whatnot. So maybe the timing does not align there. And then we do look at other available data, impact to other studies that are ongoing internally, and maybe there's a competing initiative at another institution or whatnot. We look at technical feasibility, respect to enrollment and timeline, and I really do have to highlight this as well. I know Dr. Enns said that it's very, very difficult, and I understand like your day job is really to see patients and care for patients, but timelines are very important, and enrollment as well. And then we look at, sponsor, investigator, study site capabilities, look at track records, that often comes up. And then making sure that, the site or investigator is not restricted from conducting clinical trials. And then there's this, once you do receive the grant, there are ongoing activities to monitor, including your periodic status reports. I won't go through all these in detail, but I really want to spend a little bit of time here and leave room for questions in terms of ways to help advance pediatric research. Now, I know we previously were just discussing about like the investigational product alone or the IMP, but there are many, many ways to obtain funding and also to really contribute to getting a therapy to pediatric patients. And, Dr. Enns mentioned this, but I can't underscore like how important it is to have good natural history studies so that we can understand the etiology, the range of the manifestation, and then the progression of diseases. And, FDA even now, they want longer natural history studies, not just 1 or 2 years, not just 5 years, but sometimes like 5 to 10, 15 years in terms of, if you, how the patients evolve over time. An example of this is gene therapies, right? So, that for a lot of these diseases that we're targeting for gene therapies, we don't have an idea of how they do over their lifespan. And the gene therapy studies are, it's a 1 dose only. And they initially started to go to 5-year follow-up. Now FDA has asked to go to 10-year follow-up. And, they really want to understand like the durability of benefit and also, any safety signals that can come out in the long term. So it's important, right? So some of these rare genetic diseases, in 10 years, like, do you get some sort of melanoma, right? And then so they can actually compare it when these gene therapies, like down the line, if should there be any safety signals. Another way to help contribute is understanding or conducting like qualitative patient interviews to understand the patient and caregiver journey. That's really important so that companies and regulators can understand the burden of disease from patient and caregiver perspective, not necessarily from your clinician perspective, but what does it mean for patients, right? And I think this is really interesting, like in the Duchenne muscular dystrophy population, right? So everybody always, like the clinical endpoints, we always want to preserve ambulation. But actually, from a parent's perspective, they say, you know what, actually, when my child goes into that wheelchair, he is so much less tired and he's able to move around more. So I think that's really different from kind of the developer and maybe even the clinician perspective there. And then another way to really help, and I know Dr. Enns talked about this too, is using real world data so that we can understand the epidemiology, incidence, prevalence, outcomes, healthcare utilization. How often are patients hospitalized? How often are they going to the ER? Deaths? How often they are seeing subspecialists? Understanding comorbid conditions and concomitant medication. So there are many ways to go about this, including patient registries, using insurance claims or the hospital databases to understand this. So the more information that we have, the better. And for me, honestly, I always tell my team, no, I want unbiased research. Like I don't want the company to like keep referring to ourselves if we're like citing these data. So the more unbiased research there is out there, the better. And then other things that you can do outside of research is to really, I think some of these rare pediatric diseases, we are looking for, is there a expert group? Like who are the experts and do they have consensus statements? And interesting enough, sometimes we don't have them and we really would like them. And then, contributing to clinical practice guidelines and then becoming a center of excellence. And there's two differences here. So there are clinical centers of excellence, which, based on clinical outcomes and your clinical practice, I think Stanford is really good at that. But what about being a research center of excellence? Like which sites are good sites that, actually make an effort to recruit patients to clinical trials, have been shown to contribute heavily to research. So I think this is something that is a great opportunity here. And with that, I will stop sharing and leave time for any questions. I'm sorry, there's not too much time.
That's all right. Thank you so much, Dr. Tran, for your really detailed presentation. Really love that conclusion slide as well. Any participants that have questions, please put them in the Q&A or in the chat. While you all think of that, let me share my screen. And I'd like to share with you some MCHRI resources that we do have to offer with regards to IIS studies. You all can see my screen, right? I never really know how the webinar works.
Yes.
So we compiled, thank you, Dr. Tran. We've compiled a summary document of IIS opportunities across numerous biotech companies. You'll see a screenshot here on your screen of some of the details that we're including because, like Dr. Enns mentioned, there are such different requirements across many different companies. We wanted to compile all of them into one document for you to be able to search and link to as you're looking for possible opportunities for your specific research or your specific field. We'll share this out after the webinar for you all to have access to and to look through to see what fits you best. Likewise, we will be able to provide guidance with regards to these IIS opportunities to better understand how to complete these applications, what sort of a timeline would be, if there are any questions about particular points of the application process. Additionally, we'll be able to provide warm introductions to industry contacts that might facilitate carrying out or getting feedback about your IIS applications, having events such as these to really provide you more perspectives around IIS and other opportunities to engage with industry and academic contacts. And finally, as your research really progresses, we'd like to support you in IND or investigational new drug or IDE or investigational device exemption applications to really facilitate the next step of your research. If you have questions about any of those, here is Grant and my contact information. And we're really happy to leverage the resources and the networks that we have at MCHRI to really help you advance your research to the next stage. With that, I will stop sharing my screen and check if we have any questions.
Thanks, Jessica. I do want to comment and just let everyone know that don't limit yourself to what's published, because I can tell you that oftentimes we fun things that just come through, even through like a LinkedIn message, because it depends on if it's an area of interest that you have and you see it in the pipeline, I would not hesitate to reach out to the company and just ask them for a conversation.
Great, I really appreciate hearing that. It's always reassuring to hear somebody from industry, such as yourself, really give us that sort of feedback and insight. If no participants have any questions, I think we might just wrap it right now, and I'll end the recording. Again, this recording will be posted on the MCHRI webpage for reference later, in addition to the resources that we've mentioned. I'd like to thank Dr. Enns and Dr. Tran so much for your time, all your insight, preparing such thorough slides, sharing your experiences and your perspectives. This is really such a really valuable experience. Thank you both again so much.
Thank you. Take care.
Pleasure.
Bye-bye.